All right, so we're on to kind of our last topic for the quarter, which is the chromosomal basis of inheritance. And if you look at the objectives for heredity, it's the last thing we need to do. And this week, we're going to spend time looking at it. So today we're going to look at the human genetics and disorders as far as the chromosomal basis of inheritance goes. So a little bit goes back over what I looked at yesterday and Monday, which is always a good thing, about non-disjunction and things like that. But the other parts go over something new that's called gene linkage, which is going to be the last thing that we're going to work on, you know, kind of Wednesday through Friday this week to end the quarter. So, human chromosome disorders. So, when we talk about genetic disorders, there's kind of two categories that they fit in. They are either a chromosome disorder, which means that a large part of the chromosome is changed or different, inserted or deleted or something like that. Or it might be a gene mutation where only one gene or a very, very, very small part of the DNA is affected. Now, when we come back during third quarter, we're going to talk more about gene mutations. But since one of the objectives is to understand the chromosomal inheritance of heredity, we're going to talk about chromosome disorders today. So this is where a macro section of DNA, a whole piece of a chromosome, is changed. Some of them actually have a high frequency in humans, and those occur when those occur, most of the embryos that have them are spontaneously aborted. They don't have the correct instructions to be able to develop properly because too much of their genetic information is changed. They usually cause developmental problems that result from different biochemical processes not being in place which allow the organism to survive. Certain conditions, though, are tolerated, which means that they're survivable. And these chromosome disorders lead to less of an upset in the balance of those biochemical pathways. And a lot of the times they leave the individual who has this chromosome disorder with a characteristic set of symptoms. That's what we call a syndrome. A syndrome uh, is a characteristic group of symptoms that correlate with a certain um, mechanism. So, one chromosome disorders that are usually uh, found in people that are living, so not they don't create such an imbalance that the embryo can't survive, will have a characteristic set of symptoms because of it. So non-disjunction is one of the major chromosomal abnormalities, and we talked about this one, or you did some work on this one yesterday. And it's when chromosomes don't separate properly during meiosis. But the other ones that we're going to talk about today, chromosomal mutations, are called deletion, duplication, inversion, and translocation. And these are actually pretty self-explanatory. So just by if you have an idea what those words independently mean, you can probably guess what happens to the chromosome when if they suffer from one of these mutations. So if we're looking at that second group, right? Because you had looked a lot about non-disjunction last time. All right, yesterday we had a video and you did some work with non-disjunction. Today we're going to look at these. Here is a good picture that explains these four types of chromosomal mutations. All right. In a deletion, there's a loss of a whole segment of the chromosome. So let's not say that this is one specific gene. This could be this segment D that's lost, that's deleted before and after, could have many genes on it. It could span thousands of base pairs, right? In the same way, a duplication can occur when a segment of the chromosome is repeated. And so in a deletion, you have a chromosome that's too short. And a duplication, you can have a chromosome that's too long. And both of these things affect a large enough part of the chromosome that you can see them like on a karyotype, right? And when we talk about it later in third quarter, we'll see that deletions and duplications both result from an error in DNA replication. The other two types are inversions or in translocations. So in inversion, a segment of the chromosome is actually backwards, kind of like you see compare here BCD to here DCB. It's the opposite way around. It's a segment that's been reversed. And then lastly, there are, and I think they're the most interesting, a translocation. A translocation when when a chunk, usually at the end, of one chromosome is switched with a chunk of another non-homologous chromosome. So this is the key, non-homologous chromosome. So not of the same type. And inversions and translocations usually result in errors of crossing over. So translocations can result when a crossing over occurs and two non-homologous chromosomes end up switching pieces, right? So we have pieces from chromosomes that normally should not have been recombined. So all of these can be associated with different human genetic disorders. And there's a whole host of examples for each of these. So here are some examples of human genetic disorders that are simply dominant or simply recessive. And some of them are probably pretty familiar, like Huntington's and cystic fibrosis and PKU and albinism, okay? And then two of them, achondroplasia and acromegaly, I actually talked about in one of our previous videos. So a lot of these should look pretty familiar. Non-disjunction, I know you had looked at a lot yesterday. I just wanted to make sure that we went over it again because it fits well with this section too. Just remembering that's a problem with when the meiotic spindle fibers cause errors in the daughter cells. We end up with too many or too few chromosomes. It could happen when homologous chromosomes don't separate during meiosis 1. So look here how homologous chromosomes, these two separated, but these two didn't. Or it could happen when sister chromatids don't separate properly in meiosis 2. Either way, what results is gametes that are created that either have too many, n+1, or too few, n-1, chromosomes. So here's another picture, I know, I'm sorry, I went through this yesterday that you watched, but just reinforcing this more. This is kind of what it looks like if there's error in meiosis 1 and homologous chromosomes don't separate, or if meiosis 1 occurs correctly and then meiosis 2 happens, and sister chromatids, look here, don't separate correctly, and you can see the results that when there's a non-disjunction in meiosis 1, all four sets of gametes will either have too many or too few chromosomes, where if the error occurs in meiosis II, you will have half of the gametes that still have a correct haploid number. Most of the time, non-disjunction errors that create viable offspring will result in either trisomy, so a cell having 3 copies of a chromosome, or monosomy, when cells only have 1 copy of a chromosome. That's usually what's going to happen. So, for example, Down syndrome is the result of trisomy 21, where there's 3 copies of the 21st chromosome. So a non-disjunction event took place, and the embryo ended up with an extra 21st chromosome. So its frequency is actually about 1 in 700 for children that are born in the US. And one of the issues with why this is one of the most prevalent non-disjunctive disorders is because chromosome 21 is the smallest human chromosome. But even though it is the smallest human chromosome, doesn't mean that the genetic information that's on it is any less important for development, because there's still pronounced effects about Down syndrome. Um, and the frequency of Down syndrome correlates roughly, and it's not a causation, it's a correlation with the age of the mother. So usually trisomy 21 becomes more frequent with increased age of the mother. So if we create kind of like, and this is just a very simple line graph, where we explore the incidence of Down syndrome within these age groups of the mother, you can see how it is those two variables are correlated. Kind of going in a exponential growth curve here. One thing that you might not have realized, though, is that non-disjunction can also occur with the sex chromosomes, because the human development is really more tolerant of a wrong number in sex chromosomes, having more than 2 or less than 2 sex chromosomes. But there's a variety of different syndromes that can result, and you can see some of them here, about having um, a different number of sex chromosomes. So you have 2X's and a Y. You could have 3X's. You could have an X and 2Y's. You could have an X and nothing else. All right. One of the questions is, could you just have a Y? And the answer is no. That is not viable, because the information that's on the X chromosome is critical for the development of an embryo. So it is, just like you see in this Punnett square, non-viable to only have a Y chromosome. But one of the things is that when we look at the biological, if you want, biological definition of sex, this is where things get really interesting, because you can have 2X's and a Y, you can have an X and 2Y's, you can have an X and nothing else, and you know, even the texts assign sexes to these. Is that correct? That's something that's extremely debated, even in the scientific community right now, is that, well, if you have 2X's and a Y, or if you have an X and 2Y's, what 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 are the criterion of assigning certain labels to certain genotypes? And I think in general, that's a good question to ask, not just for those who have an extra sex chromosome or a missing sex chromosome, but just in general. Genetic testing routinely occurs for non-disjunctive and chromosomal abnormalities, especially among individuals who have a familial history of such things. Amniocentesis is one of the most common methods, and it occurs in the second trimester, where a sample of the embryonic cells from the embryonic fluid are collected by inserting a hypodermic syringe kind of thusly into the uterus and pulling off some fluid and fetal cells from the amniotic fluid. And the chromosomes from there are used to make a karyotype or a picture of the chromosome like this that can be analyzed for um, non-disjunctive events or those chromosomal mutations like inversion and duplication and deletion and things like that. And then kind of the last chromosomal thing I want to talk about is polyploidy, which happens when eukaryotic cells have more than a diploid number of chromosomes. We'll talk more about this actually towards the end of the third quarter when we are looking at evolution, but it's actually a major method of evolution and speciation in plants. So as the number of chromosomes increase, so does the possible cell size. So this is kind of why you can get like huge giant strawberries. So in plant community, it's actually really, really common, but in animals not so much because it's often pretty lethal. And then just the last topic for today, and we're going to go over this a whole bunch tomorrow, as gene linkage. So genes found on the same chromosomes are considered linked. Think about sex linked. That means a gene is found on the sex chromosome. So if two genes are found on the same chromosome, they're really likely to be inherited together. Those two alleles that are found on the same chromosome, are on the same physical chromosome are more likely to be inherited together. So we can actually figure out from that a pattern of inheritance that they're the most likely to be inherited together, how close or far away they are from one another. So when genes are found on the same chromosome and they're linked, the only way that those two alleles or two genes would be inherited separately is if they were separated by a crossing over event. All right, a crossing over event. And crossing over events aren't very common, but the closer those two genes on a given chromosome are to one another, the less frequently they would be separated from crossing over. So if you can find the frequency that crossing over events happen to separate those two genes that are on the same chromosome, you can make an estimation about how closely they are located to one another on the same chromosome. And that's the earliest method for making linkage maps. And we're going to make some linkage maps on Thursday, but I'm just introducing this to you right now. So here's an example of a picture of two genes that are linked. So let's look at, here are two homologous chromosomes. These two genes, A and B, are on the same chromosome, the same physical structure. Look at how the dominant allele of A and the dominant allele of B are located on one chromosome, and the recessive allele of A and the recessive allele of B are found on the other chromosome. So, most commonly, without crossing over, the only possibility you could inherit is a dominant A and a dominant B, or a recessive A and a recessive B. It would not be very likely for you to inherit the heterozygous genotype because the two dominants are on the same chromosome. If crossing over were to occur in the region between the dominant and the or between the gene A and gene B, you could potentially have a heterozygous genotype. When crossing over occurs and separates genes that were linked, we call those products recombinant gametes or recombinant chromosomes. Since crossing over doesn't happen frequently, and since it would be even less frequent that crossing over would occur excuse me, in such a way to separate these specific genes, the recombinant gametes would happen very, 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 very few and far in between compared to the normal parental gametes like this. So here's an example of meiosis that occurs with two linked genes with no crossing over, and here occurs with crossing over. And the thing you need to remember is if you would perform like a cross where you have two individuals, you cross and make offspring, you would expect the vast majority to have one of the two, or to end up, not one of the two, but end up with inheriting chromosomes that have the genes linked like their parents. A high frequency, 80–90% of the offspring would look like that. And there would be a few that had the recombinant chromosomes. And we could look at the frequency in which recombinant chromosomes occur in the offspring to determine how far those genes are away from each other, because the more recombinants, the farther they are. The fewer recombinants, the closer they are together. All right. Now, I know this sounds kind of confusing today, and we're going to spend most of our Zoom session tomorrow going over how to do some work with this, how to figure out recombination frequency and to figure out distance. All right, but that's going to be it for today.